And Colleen, I saw your ears perk up when you heard cord blood. So I know your respective companies use stem cells derived from umbilical cord blood. And so following that first question, and I know you're a proponent of cord blood as a starting material. So following that first question, what went into this decision? And to you, what are the advantages of using cord blood as a starting material?
Okay, well, thanks for having me. And it's a pleasure to chat with my colleagues and everyone out there, and hoping we get lots of questions. So I think, honestly, part of the answer to that is really how my career started. I'm a stem cell transplant physician by training, and I started working on stem cell expansion in an area that I felt was a huge unmet need, and that was specifically in the area of patients who didn't have a donor for transplant, and cord blood could therefore be utilized. But this was back in the early 2000s. So cord blood at that point was still very, new, and patients were having very long periods of engraftment. So, sorry, my goal from the very beginning was to generate expanded numbers of rapidly repopulating cells, and that's where I started, was with cord blood. And since that time, really, we have tried other sources of stem cells, meaning, adult-derived primarily. We have not used iPSC, but we've used other sources of hematopoietic stem cells. So my companies really focus on hematopoietic-derived lineages. So cord blood really has proven to be the best source of cells for us for a number of reasons. We are no longer in the area of stem cell transplant, so I'm just going to put that in the parking lot. But cord blood really represents, as I see it, the youngest and healthiest source of starting hematopoietic stem cells for any of our stem cell-derived therapies. We don't have to modify them at all to generate our products. Cord blood, as many on the call or on the webinar will probably know, but cord blood is the only source of FDA-regulated starting material, right? So in the eyes of the FDA, cord blood is a drug. And so from our vantage point, because we are a developer of cord blood-derived cells, either expanded stem cells or immune effector cells, that starting material is critical for us. It comes, already with the FDA stamp of approval. So cord blood also really is considered to be naive and protected against all of these viral infections that adult donors have to be screened for. So it's an easier source to start with from that perspective. And it's incredibly, incredibly proliferative in our hands. So when we try the same things in our expansion system using adult-derived cells, we just do not get the same output. So this is a long way about saying, we believe that cord blood offers us a regulated source of starting material that's already FDA approved. These banks literally have BLAs, right, for their starting material. We can scale because we do pooled donor manufacturing. So this is the other reason we can take 10 units of 10 donors, cord blood donors, pool them and make our products. And this makes it very easy. So that's really why we've stuck with cord blood derived stem cells. And I love to hear that Catherine's using cord blood for iPSCs. It's a really cool, a lot of the banks, I mean, for those of you who don't know this, right, these donors are already screened, and that allows iPSC developers to select donors on a specific criteria.
Great, Colleen, thank you. Catherine, do you agree with everything in the stem cell, in the cord blood category?
I do agree with a lot of what Colleen's mentioning here. I think that the difference for BitBio is really our internal technology, which solves a lot of the problems that Colleen's mentioning. We've seen a great ability to scale up with our technology, and also the reprogramming. The technology that we use allows us to generate any kind of cell type that we'd like from nearly any source. So we've seen that possible with adult donors as well.